Hello students, welcome to EPG Pathshala. I am Satish Manchanda from Delhi Institute of Pharmaceutical Sciences and Research, New Delhi. Today we are going to discuss about a module that is characterization, application and recent advancement of nanoparticles under the paper Novel Drug Delivery System 2. So students, let's see what we are going to learn in this module. That is, first of all, we are going to learn about the introduction of nanoparticles. Then we will learn about the characterization of nanoparticles, and finally we are going to learn about the applications and recent advancement of nanoparticles. So let's start with the introduction. The question arises, why nanoparticles? Nanoparticles are intensively studied because of their possible effects on the human health. They provide numerous benefits over the available conventional dosage forms, for example tablets and capsules. The benefits they offer are greater surface area, greater bioavailability, they offer less toxicity, and they are so tiny that their removal by macrophages is avoided. Also, they are able to avoid the renal filtration due to their tiny size. They can easily cross the cell membrane, they can interact with the cell surfaces, and the targeted drug delivery to specific organs can be easily achieved. Also, they provide good retention, less clearing and enhanced bioavailability. And most of the nanoformulation prepared are biocompatible. Now, let's go to the types of nanoparticles. First of all, solid lipid nanoparticles, they are also termed as SLN. They are mainly comprises of lipids that are in solid phase at the room temperature and surfactants for emulsification, and their mean diameter ranges from 50 nanometer to 1000 nanometer. Now next is liposomes. Liposomes are vesicular structures where hydrophobic lipid bilayer surrounds an aqueous core. Next is nanostructured lipid carriers. They are also termed as NLC. They are produced from the blend of solid and liquid lipids, but particles are in solid state at body temperature. Next type is nanoshells. Nanoshells are spherical cores of a particular compound surrounded by a shell of outer coating of thin layer of another material, which is a few nanometer, that is 1 to 20 nanometer thick. Next type of nanoparticle is quantum dots. The quantum dots are semiconductor nanocrystals and core shell nanocrystals containing interfaces between different semiconductor material. Next type is supermagnetic nanoparticles. Supermagnetic molecules are those that are attached to a magnetic field but do not retain residual magnetism after the field is removed. Nanoparticles of iron oxide with diameter in the range of 5 to 100 nanometer range have been used for selective magnetic bioseparations. Next type is dendrimers. Dendrimers are unimolecular monodisperse micellar nanostructures around 20 nanometer in size with a well-defined, regularly branched symmetrical structure and a high density of functional end groups at their periphery. Let's start with our next topic, that is characterization of nanoparticles. Before going ahead for preclinical or clinical studies, it is always a sensible approach to characterize the delivery systems for various in vitro attributes through which biological behavior of nanoparticles can be predicted, thus can reduce the requirement of time-consuming and costly in vivo evaluation. Now, let's start with the in vitro characterization. Some major attributes of nanoparticles which have been considered worthy to be analyzed before conducting in vivo performances of systems are, for example, size and size distribution, shape, surface area, surface morphology, surface chemistry, drug loading, state of dispersion, physical and chemical properties of nanoparticles. Now let's start with the particle size and size distribution. Most of the properties of nanoparticles used for drug delivery depends upon the particle size and size distribution, and the methodologies commonly followed are: number 1, photon correlation microscopy or spectroscopy. Next, scanning electron microscopy, which is also termed as SEM. Next is transmission electron microscopy, that is termed as TEM. Next is laser diffractometry and atomic force microscopy, that is termed as AFM. Next is Surface charge. The charge on the nanoparticle formulation is also important parameter because it decides the interaction of nanoparticles with the biological surface. Also, the surface charge on the nanoparticles is determinant of the aggregation of nanoparticles. The techniques used for measuring surface charges are: number 1, laser Doppler anemometry, which works on the principle of laser scattering. Next is zeta potentiometer, that is based on the principle of electrophoretic mobility. Next topic is shape and surface morphology. Shape of nanoparticle is a very crucial parameter, as various studies have demonstrated the shape dependent distribution of nanoparticles in animals. Shape and surface morphology of nanoparticles can be determined by the following techniques, that is transmission electron microscopy, which is termed as TEM, scanning electron microscopy SEM, and atomic force microscopy, that is AFM. Next is entrapment efficiency and drug loading. Nanoparticles are synthesized with the aim of providing sufficient amount of drug at the site of action. Therefore, it becomes necessary to determine that how much of the drug has been encapsulated or entrapped in the core of the polymer. Now, entrapment efficiency. It is calculated with respect to the initial amount of the drug added during the formulation development. It can be determined by the direct or indirect method using the following formula, that is percent drug entrapment is equal to total drug minus free drug divided by total drug multiplied by 100. Now, drug loading. It is calculated with respect to the formulation, that is amount of the drug present in the final formulation. It can be determined by the direct or indirect method using the following formula, that is percent drug loading is equal to total weight of encapsulated drug into the formulation divided by total amount of the final formulation into 100. Now come to the direct method. Nanosuspension is subjected to centrifugation followed by washing of the pellet and subsequently lyophilization of the formulation. After that, lyophilized particles are dissolved into the common solvent in which both the drugs and polymer are soluble, and then samples are subjected to analysis for drug content using suitable analytical technique. Now come to the indirect method. Under this, nanosuspension is subjected to centrifugation followed by analysis of drug content into the supernatant using suitable analytical techniques. Surface hydrophobicity. Hydrophobicity of nanoparticles influences the interaction of nanoparticles with the biological system and ultimately influences the biofate. Surface hydrophobicity of nanoparticles is measured by: number 1, water contact angle measurement, dye binding, that is rose bengal dye. Next, hydrophobic interaction chromatography and X-ray photon electron microscopy. Carrier drug interactions. Interaction studies of all the components of formulations are done by DSC, that is differential scanning calorimeter. It is a preliminary technique for the identification of any possible interaction between formulation components. Thermograms of each constituent of the formulations are recorded and compared with that of final formulation. Appearance of any extra peak or disappearance of existing peak shows the interaction among the components. Next technique is FTIR. It is a confirmatory analytical tool to define the interaction between formulation components. FTIR spectra of each constituent of the formulation are recorded and compared with that of the final formulation. Appearance of any extra peak or disappearance of existing peak shows the interaction among the components. Physical state characterization. Physical state of the drug greatly influences the solubility and dissolution profile, and consequently release profile and in vivo activity. Hence, it is worthy to determine the physical state of drug after encapsulating into nanoparticles. It can be determined by the following techniques. Number 1 is DSC. Drug in the crystalline state shows a sharp and symmetric DSC thermogram, while it becomes broadened and asymmetric in case of amorphous state or may disappear if the drug turns into molecular state or solid solution as usually observed after encapsulating crystalline drug into nanoparticles. Next technique is XRD. Powder X-ray diffraction may be readily used to determine the crystal structure of simple lattice structures. In this technique, diffraction of X-rays at a specific range of angle is observed. The crystalline substances give sharper peaks due to their crystalline symmetry, unlike amorphous ones. Nanoparticle dispersion stability. For this, critical flocculation temperature of nanoparticle suspension is recorded along with the zeta potential, which gives estimate of the surface charge and ultimately the coagulation tendency of particulate system. Next is surface area. BET, small angle X-ray scattering, and nitrogen absorption method is commonly used for it. Next parameter is release profile. It is necessary to determine the release pattern of the nanoparticle formulations, whether they are releasing the drug in the desired manner or not, and studied by dialysis method, diffusion cell, etc. Release kinetics. Release kinetics is determined by fitting the release data into various kinetic models, including zero order, first order, Higuchi, and Korsmeyer-Pepys model. These models establish the relationship between cumulative release versus time. They also reveal the release pattern, that is, release is based on Fickian or non-Fickian diffusion phenomena. Next is ex vivo study under the headings of in situ studies. In science, ex vivo refers to experimentation or measurements done in or on tissues from an organism in an environment mimicking natural conditions. Ex vivo conditions permit experimentation on living cells or tissues under more maintained conditions than is possible in vivo experiments. For example, Barberi et al. In year 2014 studied nanoformulations based on phospholipid and chitosan for tamoxifen, that is an anti-cancer drug which is slightly soluble in water. They investigated the permeation of tamoxifen released from prepared nanoparticles in an using chamber on which excised rat intestinal wall was mounted and improved drug permeation through the rat intestinal tissue by the mechanism of paracellular transport. Next example is Baer et al. In the year 2010 prepared nanoparticles by the polyelectrolyte complexation, that is PEC method for colon delivery with the insulin loading more than 80%. On performing ex vivo investigations on colon membrane of rats, it was found that better insulin transport occurred by formulations made with quaternized derivative. Also, in the year 2010, Sana et al. Prepared nanoparticles by oil in water high shear homogenization. They use pericyrol and fatty alcohols as lipid phase. After incorporation into hydrogels, ex vivo drug permeation tests were investigated across porcine stratum corneum and concluded that the drug release from formulations having fatty alcohols increases as the alcohol chain length increases. In vivo characterization. In vivo studies include the study in which several biological entities are tested on living organisms, usually animals, including human and plants. The selection of living systems for in vivo studies depend on the therapeutic category of drug, type of formulation, and desired site of action of the drug. Before any experimentation, protocol for research study is submitted to Institutional Animal Ethics Committee for new experiments or extension of ongoing experiments for animals other than the non-human primates. Basically, it involves two types of studies, that is pharmacokinetic studies and pharmacodynamic studies. Pharmacokinetic studies. These type of studies basically involve the measurement of blood drug concentration at different time points. Since nanoparticles show different disposition pattern in comparison to naked drugs. Thus, nanoparticles are capable to alter the fate of drug, that is Cmax, Tmax, AUC, MRT, AUMC, volume of distribution, clearance, biodistribution, etc. and consequently pharmacokinetic profile. Pharmacokinetic studies involve the measurement of set parameters while biodistribution studies involve the quantitative assessment of drug into the body organs as well as targeted organs. Pharmacodynamic studies. Pharmacodynamic studies actually involves the assessment of in vivo activity, that is, the evaluation of potential of drug-loaded nanoparticles against the disease state in suitable animal models and comparison with their already existing formulation. For example, anti-inflammatory drugs are evaluated against rat paw edema model. Anti-diabetic drugs are evaluated against streptozotocin-induced diabetic condition. Anti-cancer drug nanoparticles are assessed for tumor inhibition potential against tumor-bearing mice, while immunization involves the measurement of antibody. Some examples of in vivo evaluations are, for example, Tardrus et al. In year 2015 assessed safety as well as pharmacokinetics profile of CHL-loaded lipoprotein mimic nanoparticles through intravenous administration. The dispersion of nanoparticles was studied for in vitro hemolytic ability, in vivo vascular irritation studies in rabbit, and in vivo pharmacokinetics after intravenous administration in rats. Next example include Zhou et al. In the year 2015 prepared chitosan-based nanoparticles for the sustained release of berberine chloride. Novel berberine chloride loaded chitosan nanoparticles were spherical in shape, exhibited good stability with good in vitro release profile. After injecting nanoparticles, the retention time in synovial fluid increased and the level of drug in red plasma decreased. In vivo evaluation showed higher anti-apoptosis activity. The next example include Abrego et al. In 2015 developed tranoprofen-loaded PLGA nanoparticles dispersed in hydrogels for preparing semi-solid formulations for ocular administration and performed ex vivo and in vivo studies. The results of ex vivo corneal permeation and in vivo anti-inflammatory efficacy studies included that the ocular application of the hydrogels with ozone was more effective as compared to ozone-free formulations for treating edema on the ocular surface. Ocular irritancy was not observed for the hydrogels. Another example includes Sina et al. In 2013 developed polylactide co-glycolide nanoparticles containing voriconazole by multiple emulsification process and also made them porous by presence of an effervescent mixture for better pulmonary delivery. Pulmonary deposition of the nanoparticles was analyzed by customized inhalation chamber. The drug was found in lungs for 7 days in case of porous and for 5 days in case of non-porous after administration. Nanoparticles with enhanced drug loading were delivered to murine lungs. Lower mass median aerodynamic diameter having porous nanoparticles had good pulmonary deposition and presence in lungs for a longer duration. Applications and recent advancement of nanoparticles. The primary goals for research of nanoformulation in drug delivery include number 1, delivery to targeted organs, number 2, therapeutic efficacy with minimum toxicity, greater safety and biocompatibility, and finally faster development of new safe medicines. Application of nanoparticles include, first of all, nanoparticles in medical imaging. Under this, optical imaging. Quantum dots are nanoparticles composed of inorganic semiconductor molecules which emit fluorescent light under UV illumination. Thus, disadvantages of conventional dyes or fluorescent compounds are avoided by using quantum dots. Next is magnetic resonance imaging. The relaxation time of supermagnetic nanoparticles such as iron oxide are much higher than those of conventional gadolinium-based agents. Nanoparticles in targeted drug delivery. Treatment of a disease of specific organ is not possible without affecting the other organ by the conventional drug therapies. The conventional drug therapies show their effect on other organs, due to which a patient may suffer from a new disease before getting treated for the previous one. Therefore, the concept of targeted drug delivery system emerged and has been successfully achieved by nanoformulations. Nanoparticles in targeted drug delivery for cancer. Paclitaxel is a well-known anti-cancer agent which is water insoluble, was loaded within nanoparticles of albumin. This soluble form of paclitaxel has eliminated the side effects but also improves transport of the drug from bloodstream to the tumor site. The US brand for this is Abraxane. Another example include Kilike et al. In 2011 studied nanoformulation of PHBHHX, which is a polymer named poly 3 hydroxybutyrate co-3 hydroxyhexanoate, which is a natural polymer loaded with an anti-neoplastic agent using etoposide and folic acid as ligand. The cytotoxicity of conjugated nanoparticles to the cancerous cells was observed on higher side than that of normal fibroblast cells, concluding that the conjugated nanoparticles have the capability for selectively targeting cancer cells. All formulations of the PHBHHX nanoparticles were evaluated for apoptotic or necrotic activities and also tested cytotoxicity. The results obtained concluded that the folic acid attached PHBHHX nanoparticles of etoposide exhibited promising results for the targeted cancer therapy. Another example include Albieli et al. In 2015 focused on designing biocompatible magnetic nanoparticles for magnetic targeted drug delivery, also termed as MTD. Doxorubicin-loaded magnetic gold nanoparticles functionalized with thiol-terminated polyethylene glycol were prepared. In vivo studies proved that a higher accumulation of drug at tumor site can be achieved by magnetic targeted drug delivery as compared to the drug accumulation obtained by passive targeting. Neurodegenerative diseases. The selective nature of the CNS organ is a biggest problem in achieving the localized drug action for the neurodegenerative diseases. The attempts have been made to cross the selective barriers of the CNS by different approaches, and nanoparticles come under the successful attempts. For delivery of drugs in brain, blood-brain barrier, that is BBB, is the toughest hurdle. Nanoparticles of polyhexadecyl cyanoacrylate, that is PHDCA, and related compounds have been shown to facilitate drug transport across the blood-brain barrier. The example of neurodegenerative diseases include the study of Calvo and colleagues in the year 2001 and 2002. They synthesized a nanoparticle system composed of a copolymer of PEG and PHDCA. PEG is not recognized as a foreign material by macrophages in blood and can therefore increase the half-life of the drug carriers in blood, as per the studies of Harris and Chess in the year 2003 also. Another example include the example of liposome. In the year 2003, Skimt and colleagues prepared liposomes with diameter ranging from 90 to 100 nanometer to encapsulate prednisolone for multiple sclerosis treatment. Another example include the delivery of gene. A further application of PEG conjugated liposome is in gene delivery across the blood-brain barrier. Shai et al. In the year 2001 delivered plasmid DNA encoding beta-galactosidase across the blood-brain barrier in rats. Ocular diseases. The main drawback of conventional ocular dosage form is nasolacrimal drainage, systemic effects, dosing frequency, etc. The ocular nanoparticles are designed to prolong drug resistance time by trapping the drug in the ocular mucus layer. In the year 2002, Pignataro and colleagues have used commercially available udrazit polymers to deliver non-steroidal anti-inflammatory drugs, that is flurbiprofen and ibuprofen, to rabbit eyes. Another example include study of Chonker et al. In 2015, who reported amphotericin B loaded lecithin chitosan nanoparticles for ocular application for prolonged effect. Nanoparticles were found to have antifungal susceptibility against Candida albicans and Aspergillus fumigates and improve bioavailability approximately twofold and improve the precorneal residence time. Application of nanoparticles in HIV and AIDS. D. Jangri and colleagues in the year 2000 investigated the delivery of an HIV-1 protease inhibitor CGP70726 by nanoparticles made from a copolymer of methacrylic acid and ethyl acrylate, that is Udragid L155. The HIV-1 TAT protein is a prophylactic or therapeutic vaccine against HIV-1 AIDS as per the study of Cafaro et al. 1999 and Caputo et al. 2004. Rudolph et al. In the year 2004 recently described an SLN based system consisting of DNA compacted with a TAT protein. Application of nanoparticles in respiratory diseases. The conventional treatment is associated with number of systemic side effects, and hence respiratory diseases demand more specific and targeted action of drugs, which has been fulfilled by the use of nanoparticle formulations. The example include in the year 2003, John et al. Demonstrated the use of a liposome-based nanoparticle system to inhibit inflammation in a murine model of allergic asthma. The strategy employed was to inhibit P-selectin receptors on activated endothelial cells in circulation, which reduces interactions between endothelial cells. Another example include the study of Fang et al. In the year 2015, who developed a novel nanoformulation termed phosphatosiomes with an aim to improve the targeting capability of phosphodiesterase 4 inhibitor to the lungs for the treatment of acute lung injury by intravenous administration. Phosphatidylcholine, that is PC, and distereoyl phosphatidyl inolamine and polyethylene glycol were used for synthesizing phosphatiosomes and loaded with rolipram, which activated neutrophil inhibition. They also studied the possibility of using phosphatiosomes as the lung targeting carrier, and acute lung injury treatment were performed. Application of nanoparticle in gene delivery. The drug delivery system for the alteration in genetic makeup for treatment of a disease was a kind of dream before the development of the particulate system of nanoparticles, because reaching to the one of the basic unit of living system is quite impossible unless until the size of the system is very less. The example include in the year 2007, Swami et al. Developed a nanoparticle drug delivery system composed of 1,4-butanediol diglycidyl ether, which is bisapoxide, which was cross-linked with polyethylamine. The developed system, that is nanoparticle DNA complex, was found to show 2.5 to 5-fold gene transfer efficiency and very less cytotoxicity in cultured cell lines as compared to the conventional methods used for gene transfer, for example, lipofectamine 2000 and fusin. Oral bioavailability enhancement. A number of research groups has demonstrated improvement of oral bioavailability of drugs, which elicits poor oral bioavailability. For example, Joshi et al. In 2014 demonstrated approximately 18-fold improvement in oral bioavailability of gemcitabine. Furthermore, Kalaria et al. In year 2009 and Tariq et al. In 2015 reported approximately 4-fold enhancement oral bioavailability of doxorubicin and epirubicin respectively through polymeric nanoparticles. Oral immunization. Oral immunization is always preferred over systemic one owing to following inherent advantages. That is, enhanced patient compliance, they avoid the risk of pain and trauma, they are economic, and they eliminate the risk of needle-borne infection. At present, it has shown great potential for including mucosal immune response to fight with pathogen at entry site. However, instability of the antigen in the hash GI milieu as well as poor permeability across GI membrane limits the possibility of oral vaccination. However, nanoparticles have shown the ability to overcome the aforesaid drawbacks. In the year 2015, Hardy et al. Demonstrated significantly higher humoral, mucosal, and cellular immune response for tetanus toxoid loaded stable glucosaminosylated chitosan nanoparticles after oral administration, while commercial TT vaccine was found unable to elicit any type of immune response after following oral administration. So students, let's now summarize what we have learned from this module. Nanoparticles are gaining attention of formulation scientists on a regular basis, which is also enhancing the standard of drug delivery approaches. Next, to ascertain the efficiency of nanoparticles, a number of properties needs to be checked or analyzed so as to decide the fate of nanoformulation in the living systems. The characterization of nanoparticles consists of in vitro, ex vivo and in vivo studies on the basis of which formulation scientists take decision regarding the behavior of nanoparticles within the human body. Next and the final is, therefore, nanoformulations should be carefully characterized and the methodology adopted for characterization must be accurate and should not compromise with the quality of results. Thank you.